Hello friends, in the last video we had discussed about the we have discussed about this unwinding of DNA and the leading strand and the lagging strand and everything, and how the RNA template is used as the primer for this DNA polymerase, and primase is the enzyme that synthesizes this RNA template. And now uh, we will talk about certain things, two enzymes, topoisomerase 1 and 2. In this case, let me take a new color. uh, this is the helicase enzyme that is running ahead of the replication fork and uh, uh, opening the double helix, which is joined by the hydrogen bonds here. And as it moves, the the fork, as the fork moves, the bond opens, but it but ahead of this fork, the DNA starts to coil much farther, which is called a supercoiling. Like if suppose two strings are joined and you try to open this thing, you pulling these two ends on other two parts, it will further increase the coiling over here. So this increase of coiling is called supercoiling, and it exactly happens with the DNA as well. So when this helicase enzyme is moving upwards along with the replication fork, the more supercoiling is increased in this DNA molecule. So it may cause some deformity in the DNA structure. It may break the phosphodiester bond. So in that case, to maintain the to maintain the to maintain the integrity of the DNA molecule, to enzyme works here, that one is the topoisomerase 1 and another one is topoisomerase 2. And these two enzymes more or less do the similar kind of a function, but there are definitely some difference. In case of topoisomerase, suppose if the supercoiling is like this, and if when the topoisomerase functions, topoisomerase 1 function, and when it is the supercoiled area, one of the strand breaks. So if, let me take another color, one of the strand breaks here, suppose, and it unwinds from the other strand, decreasing the supercoiling. And so and again it ligates the strand. So the supercoil has been removed to maintain the integrity, but at the same time, the the single strand break, which is done by this topoisomerase 1, they again get ligated by its own function. Now this topoisomerase 1 is of two, there are two types, like topoisomerase 1A and topoisomerase 1B. This 1A remains associated with the 5' end after the cleaving, and in case of this topoisomerase B, it remains associated with the 3' end after the cleaving. Okay, now this there are some informations that I would like to give you, is this, that in case of E. coli, uh, this topoisomerase 1 removes both the negative supercoiling. It, in case of E. coli, this topoisomerase 1 removes the negative supercoiling. Okay, and in case of cuff thymus, uh, this topoisomerase 1 removes both the positive and the negative supercoiling. This positive and negative actually defines the direction of the supercoiling. And now in case of this topoisomerase 2, this topoisomerase topo 2, you can write it in this way. Topoisomerase 2 actually breaks the double strand and it releases to DNA molecule. This kind of things happens in the theta replication. We can see this kind of replication in the prokaryotes where the uh, structure of the DNA is circular, and this is the structure. This is double stranded structure. I'm not really good in in drawing this kind of a circle. So what happens when the replication bubble is produced, it is like this. Alright, so now when the replication bubble is produced, these are the two forks. One moves in this side, another moves in that side, and so two two farther to a circular DNA is produced, but they remain interlocked to each other. This is the structure of this interlocked DNA of after this theta replication. And to release the one molecule of DNA from the other, this topoisomerase 2, this topo 2 functions here, and it breaks double strand of one of a DNA molecule. So let me take another color so it will be easy for you to understand. It breaks one of a bond. Okay, suppose here, and then what happens is this, that one of the DNA molecule remain intact, and the other one which is broken, it again is again ligated, and a complete circular double bond is now there. So from this single parental DNA molecule, after this theta replication, when two concatenated or interlocked DNA molecule is produced, then this topoisomerase 2 functions. It breaks two of the strands of one of the DNA molecule, and it releases it from the other, and now there are two daughter DNA molecule. So this is the function of the topoisomerase 1 and topoisomerase 2. Now this there is another enzyme called Gyrase, which has got a very similar kind of a function of like this topoisomerase, and it breaks the phosphodiester bond and link it again. So this is just similar, like this Gyrase is working similarly to this topoisomerase enzyme, and it is made up of two heterotetrameric subunits, like these are made up of four subunits. That's why it's a tetrameric, and it is heterotetrameric. Why? Because there are two subunits, A and B. So the subunit Gyrase A and Gyrase B is involved, and these are in two copies. And this Gyrase A is responsible for cutting and joining. This is responsible for cutting and joining, and this whole thing is done in the presence of ATP. So this Gyrase B hydrolyzes ATP to get the energy to do this kind of a function. So now till so far we have understood the replication initiation, elongation, and then this how to release the supercoiling, and how to open up the interlocked DNA molecule. But this all discussion was on prokaryotes, and now we will be starting our discussion on the eukaryotes. So take another whiteboard, and here in case of eukaryotes, well, there are some more this regulation of the DNA replication is much more compact, and some more proteins are involved in it. So in case of this DNA replication origin, this study was done in yeast, and this replication origin is called ARS, that is the autonomously replicating replicating sequence. Now this sequence again is 200 bp long, which is much shorter than the E. coli replication site of origin, and this is also an AT rich sequence. Basically all these DNA replication start sequence are AT rich because, as we know, that A and T are bonded with two hydrogen bonds, and G and C has 3 hydrogen bonds in themselves. So in case of opening the DNA double helix, it is much more easier for AT to break down. So in case of those places where we need to open up the DNA double helix are rich in AT. Okay, so this region contains an A domain, and another one is B1 to B3 domain, and these domains are also this domain contains this A domain basically contains an 11 BP consensus sequence consist of 80 rich base pairs, and I'm lacking of time, so I will be discussing in details in my next video, so stay tuned. Thank you.